Questions on the exam, again, please feel free to email those to me. I'm posting everything on the discussion groups in D2L about that. No, I do not have your Scantrons. The reason for that is someone walked off with the alphabetizer from the biology office, and I need to now do all that by hand, which takes considerably longer. Or I could just like throw all of them out in the room and you could grab them. That would be a possibility too. I think that wouldn't work quite as well. Clicker questions. Again, please email me your clicker ID and Odin ID if you have 0 points and no clicker that's attached to them. If you have fewer points than you think you should have, don't email me. If you do, I'm just going to ignore your email. Okay, so speaking of clickers, let's go ahead and have today's first. I've switched them now. We're going to call them 10.1 and 10.2. It makes it easier for me to find these particular clicker numbers. So given what we've discussed in class, we had a mutation in topoisomerase 1 in E. coli that inactivated it. Assume there's only one and that everything else in the E. coli genome is normal. You would expect A, genome replication not to take place, genome replication to happen normally, genome replication to take less time than normal, genome replication to take more time than normal, the cells to replicate their genomes but not divide. Again, feel free to discuss. Ten. 5. Okay, we have some differences of opinion on this one. So basically, topo 1 missing in E. coli, what would happen? Not take place, happen normally, less time, more time, or they replicate their genomes but don't divide. So again, difference of opinion here. Basic message as far as it's concerned is there are 2 kinds of topoisomerases, topo 1 and topo 2. Topo 2 can do exactly the same thing as topo 1 in terms of getting rid of the extra supercoils. How are those supercoils being generated in E. coli? You're pulling apart the strands. So pulling apart strands is going to end up overwinding your DNA or making it make these funky extra loops. You can either take care of those with topoisomerase 1 or topoisomerase 2. So the cell can, in fact, survive. But it's going to take considerably longer because you've made all of these extra supercoils that it has to deal with. So the answer is D, yes, yay, for those of you who got it. Yes, question?
I'm just checking to make sure. For some reason in my mind I thought topoisomerase 1 is connected to the supercomplex, but topoisomerase 2 isn't, so that's why it would take longer?
So the question here has to do with what's present at the really replication complex, the replication fork. In those diagrams, it has topoisomerase. That topoisomerase could be a type 1 or a type 2. And so it doesn't really matter which one it is. And people will argue about which one it is under what circumstances. And it kind of depends what lab you're working with, et cetera, at the time. Yeah?
What did you mean by there's only one? Like, what did you mean by that? I was assuming that it was, one.
Right. So it turns out there are multiple type 1 topoisomerases in E. coli. We haven't talked about it, so that's why I didn't want to put it in here. But just in case we have some expert E. coli geneticists in here, I put that in there. Because it turns out there are multiple ones because it does turn out to be really quite important. You know, yes. Pardon?
You were not including topoisomerase 2 in that.
Yes. So. Yeah. So there's only one, because it says topoisomerase 1. I think that's clear from the question. I hope it is. Or some people maybe confuse some people. Sorry, if they were lacking topoisomerases completely, then what would the answer be? Yeah, then they would never be able to replicate. Yeah, it would cause all kinds of problems. Okay, make sense? Yes? No? Still no? We can always talk about it later offline. So our next question for today is, which DNA polymerase fills in the lagging strand telomeres where primers have been removed? DNA polymerase 1, DNA polymerase 3, DNA polymerase alpha, DNA polymerase delta epsilon, or telomerase? Ten, 5. Okay, looks like I did a really crummy job of explaining this. So we're all over the place, and maybe it has to do with the image that didn't show up in my lecture last time. I'll try and get to that in just a second. So why can't it be A or B? E. coli does not have telomeres, so it can't be A or B. Okay, so C, D, or E. DNA polymerase alpha does what? It extends primers, but it's just part of the primase complex. Is that really going to help filling in the piece that's gotten chopped off because of the telomere? Telomerase, what does that do? It extends the template strand. The template strand gets extended by the telomerase. We'll take a quick look at that in just a second. So the answer is D. So it's the DNA polymerase delta or epsilon, which obviously I did a bad job of explaining. So we'll try and find this in the previous lecture. Hang on just a sec, and I will pull that up. And then maybe it decided it's not going to show it there either. Yay. OK, I don't know why it didn't work last time. It's very bizarre. So what happens at a telomere? You have your end of the DNA. This is where there was a primer that the primase put in. And this primer has been removed by RNase H. So now you need to do something about this end. Otherwise, each time when you replicate, that end's going to get shorter and shorter and shorter, and eventually get into important genes. So what the telomerase does is it extends the 3' end, because it's a DNA polymerase. Just like all DNA polymerases, all it can do is extend from a 3' OH. So it keeps extending from this 3' OH using the RNA template. So you get this long, many repeats that happen here. Varies from organism to organism, from cell to cell. But now you've got usually hundreds of repeats of this particular sequence. Now you can have a primase come and bind to this single strand, make the RNA. That will then get extended by the normal DNA polymerases that are present inside the cell. Well, those will be delta epsilon. Again, people will argue about leading or lagging strand here. And then the primer will get degraded. This will extend some more. Primates will come down, extend it, primate will get degraded. So it's the filling in of this opposite strand is the cellular normal replicative polymerase. All that the telomerase is doing is making a longer template that can then be copied. So again, apparently that had something to do with this image, which again probably means towards the end of today's lecture we'll have trouble with images again too. So does that make more sense? Yes? No? Okay. How does it know when to stop? So the question is, how does the telomerase know when to stop? That is a fabulous question, and nobody has a good answer to it. And in fact, if you're interested, read those extra review articles that I put in at the end of the lecture last time. They have some ideas, but again, exactly how many copies seems to have to do with how long before a cell division takes place, et cetera. So it's a great question, exactly how many copies you get. So the rest of today, we're going to talk about how horrible DNA is as a genetic molecule. And just what we got. So the main reason that DNA is such a horrible genetic molecule is that all of the information that you have in DNA in terms of the genes, the transfer of information, is all chemical information. And chemistry happens. We're in aqueous solutions. We're at relatively high temperatures relative to the particular stability of DNA. And what that means is just by living, our DNA gets damaged. And that damage happens to do with chemical modifications. And again, the chemical modifications are those things which have the information that needs to be transferred as being part of the genetic material. So any kind of change that happens in the DNA molecule, which is a change in terms of the genetic information, that'll be a mutation that happens. Mutations, if they will make a specific change that gets copied incorrectly, and not all mutations are that way, but something which gets copied incorrectly, those will then be inherited. So if you have a particular change, and we'll talk about deamination of cytosine, which then causes a change in the base pairing, then you'll have something which can get passed down from generation to generation, or just from cell to cell. Other things that this chemical change can do can block replication, can block transcription, which clearly if you need to replicate your cells, or you need to make a copy of the RNA from the DNA, this is going to be a problem. Lots of these chemical changes seem to happen in cancers. So you've probably all heard about carcinogens. Almost all carcinogens cause a chemical change of some kind to the DNA. But at the same time, one of the best ways to deal with cancer cells is to treat them with large amounts of mutagens. So radiation therapy, chemotherapy is all about DNA damage. Yeah?
Random question, maybe not a real thing. Is it because it's, I've never thought about this, is it because it's like trying to shake the DNA back into the right position? Or like, what is it blasting it with more, mutating the DNA further or risking that? What's the benefit there?
So I'll paraphrase your question now. What's the benefit of causing a whole bunch of DNA damage? Yeah. We'll get back to that a little bit later on. It turns out there are lots of sensors of DNA damage. And those sensors of DNA damage cause then the cell to basically commit suicide. And so that's the reasoning behind using a lot of these things. Or once they've replicated and you've got all these changes that have happened, you will have mutated something that's critical for replication to happen. So that's the idea behind that. So there are really two sort of reasons that this is used for chemotherapy aspects of things. Are mutations involved in aging? This question mark here should actually be really huge because it's not terribly clear whether these particular things happen. But clearly, just the fact that you're living in an aqueous environment at a particular temperature means there are going to be lots of changes that happen in your DNA. There are a couple of things that save DNA in terms of being a decent molecule for the genetic information. Probably the most important one is the one in red down at the bottom, which says evolution. Because if you didn't have changes in the DNA, we'd all be exactly the same. So therefore, a little bit of change is good, not too much change. The other thing, which certainly in terms of DNA repair is really important, is that you have a backup copy. All the time you have a backup copy. So if something happens on one of these strands, you should have the information on the other strand to say, hey, this should be C, A, T, or G, as opposed to some funky hypoxanthine, whatever it happens to be, which ended up being there. We do know a number of cases. We talked about one already, having to do with diseases of DNA repair. Most of these are inherited diseases. We already talked about colon cancer. Many colon cancers, at least the hereditary ones, have to do with mutations in mismatch repair proteins. Xeroderma pigmentosum. This is a, again, genetic disease which people are hypersensitive to ultraviolet irradiation. And it turns out that a lot of the genes that are involved in the process of dealing with the mutations that happen due to ultraviolet radiation are these nucleotide excision repair proteins. In fact, a lot of what we know about how and what proteins are involved in dealing with this kind of DNA damage come from cells of people who have had these diseases. AT, with its unpronounceable name, I'll try it anyway, ataxia telangiectasia, is quite commonly mutated in a number of different cancers. This is getting back to your question about why do all of these changes. It's because of these particular proteins, the ATM protein, which is a checkpoint protein. And that we'll talk a lot more about in cell biology, hopefully next term when you guys are talking about it. I'll skip some of the rest of them except for this one down here, which I'm not going to talk about any more than just right now. Fanconi anemia is DNA interstrand, get you in a second, cross-link. So it's a cross-link between one strand on the DNA and the other strand on DNA. This happens relatively infrequently, fortunately, because that's clearly a problem when you're linking two strands together. You don't have that backup copy anymore. The reason I mention it is there's a couple of really good research labs up the hill at OHSU who are working on Fanconi anemia and trying to understand what the proteins are that are involved with dealing with that particular problem. Yeah, question in the back. I would not assume that you would know all of these things. I would, however, know that colon cancer, these hereditary colon cancers are involved in mismatch repair because we've talked about it at least 2 or 3 times now. Make it 4, so I just talked about it now. And that the xeroderma pigmentosum in terms of those repairs. The other things I would say I wouldn't worry about, BRCA2, which is totally cool and interesting. We may talk a little bit more about that later. But I wouldn't look at this table and say, hey, what's the most nitpicky thing I can find in here? Let's see. OK, in lymphomas, what's the most likely gene that has been mutated? I would not do something like that. At least I'll promise it now. We'll see when I write the exam. No, I'm just kidding. No, I wouldn't. I try not to do things like that if I can possibly avoid it. Is there another question here? No? OK. OK, so let's talk about some of the problems that we have. The main problems that we have, of course, is that we like it warm, oxygenated, and light. All of these things are a problem as far as DNA is concerned. There are some more major problems, and those have to do with why working in a nuclear power plant or being near an atomic bomb are problems, and this is ionizing radiation. Ionizing radiation almost always leads to some of the problems that this model has, like this thing here hanging out, backbone breakage. So the phosphodiester backbone causes those breaks, and radiation therapy, that's exactly what radiation therapy does, is it causes lots of breaks in the backbone of DNA. So yes, it's a problem if you're exposed to too much ionizing radiation, but that's the LINAC, all of these things they've got. Actually, people interested in, anybody taken the radiation physics course? Physics for radiation medicine? Fabulous course, works directly actually with the radiation medicine group up at OHSU. So if you're, all those physics classes you have to take, there is a reason for that. Chemical-based modification. This is probably the most common direct modification to a single base, and we'll talk much more about this later when we talk about methylated DNA. But The amine groups that are hanging off of all of the bases that have amines, which are, take a look at it again, but it's the guanines, adenines, and particularly cytosines. Deamination, which is the loss of that NH2, happens very frequently. And in any particular cell, it's probably hundreds per day that happen. It's just this amine group falls off. And that's just because we are in an aqueous environment, and there's a certain amount of heat and oxygen which are around. But mostly having just due to the temperature. Even more important in terms of heat is the depurination which happens. Purines are the big nucleotides. So these are adenines and guanines. They have a really wonderful tendency to fall off of the phosphodiester backbone. And that happens tens of thousands per day. So really, really frequent that these purines are falling off the DNA. Pyrimidines fall off as well, but a little less frequently. And then UV irradiation, which we talked about already, just walking outside. Now, fortunately, we're in Portland, so we don't have to worry too much about UV irradiation. But the just effects of UV irradiation on pretty much all organisms causes pyrimidine dimers to form. So pyrimidines, purines. I did not ask you a question about drawing a base pair on the exam, but I do expect you to remember that the purines are the short name, big side chain, pyrimidines, long name, short side chain. Pyrimidine dimers, these are pyrimidines that are right next to each other in the sequence on one strand. A lot of people talk about thymidine dimers, but it just happens it's that single ring structure. So you can get thymidine dimers, you can get cytosine dimers, you can get thymine cytosine dimers, and what happens is a cross-link that happens between these two. Now, since DNA has this helical structure where it wraps around, if you've got cross-linking between these two, what it does is it pulls those things together. So it really messes up the structure of DNA, and we'll see that a lot more in just a second. Yeah? Does uracil have the same issue in RNA? Uracil, the chemistry of uracil is going to be very similar as far as that's concerned, but in RNAs, you rarely are going to have those kinds of base stacking interactions like you have in double-stranded DNA. So in theory, yes. In practice, it's not so much of an issue. Okay, so let's talk about some of these mutations. A lot of this is just terminology, but this is something you'll see in the literature, in your textbooks, on MCATs, these kinds of terminology here. So transitions are just same to same, and I use the single letter code here again. I don't expect you to remember it. It's just easier shorthand for me. Y is for pyrimidines, R is for purines. So a transition is just basically sort of the same size piece that you're changing. Transitions, transversions, we'll see this when we took a look at the genetic code a little bit later on. This actually usually changes the proteins very little in terms of these changes. Turns out they're also a little bit harder to detect as far as the repair machinery is concerned. On the other hand, a transversion, which is you take a big and go to a small, or take a small and go to a big, which is shown here on the right-hand side. So adenines are changing for cytosines, or for that matter, with thymidines. These are ones which are much more likely to cause changes in your genetic code, but also are much easier to detect. Because just like the case for polymerization, where you're putting in things by detecting what the geometry is, a lot of times DNA damage is being detected by looking at the geometry, seeing if there's a change. So if you've taken a big nucleotide and swapped it for a small nucleotide, that's clearly going to change your geometry. Small, big to small, small to big, you name it. That's going to change your geometry a lot. So these are a lot easier to detect. We already talked a little bit about mismatch repair. Wanted to just sort of follow up on this because we, I think, didn't talk about it as much as we probably should have. Here, if you just have a misincorporation, and so here, the misincorporation is a transversion. You have this T here. You have a G bit put into it by mistake, some problem that's happened in the polymerization process. As long as you have appropriate mismatch repair, that will take out this G on the new strand, which is detected how in E. coli? Methylation of the new strand, exactly. probably nicks in the case of your eukaryotic replication. Nick just means a break in the backbone, by the way. Someone asked me about that after class. So you repair it. However, this does not get repaired clearly. This new base, the G, will end up getting paired with a C. And so now you've gone from an A at this position to a C at this position. What kind of mutation is that? A transversion, which has now taken place. So where are we getting these DNA damage locations? There are a couple of ways of looking at this. I've color-coded this in such a way that everything which is blue is hydrolysis, red is oxidative damage, and green is methylation. The size of the arrows here corresponds to how frequently these kinds of chemical changes leading to mutations take place. So here, big blue arrows, deamination. Not deamination, depurination. Here, purine, again, it's one of the big ones. Just chops this bond here. This base is lost. Same thing is true over here for adenine. Hydrolysis, this base is lost. Hydrolysis here for deamination of cytosine. It's the most common of these deaminations, but you also have deamination that happens on adenine, and you have deamination that happens on guanine. Oxidative damage, the most common oxidative damage that you have is at this position right here, the 8 position in the ring, either in terms of guanine or adenine. We'll talk mostly about the guanine oxidative damage that takes place. You can also have methylation, happens a lot less frequently, but you can also have methylation that takes place. Another way of looking at this is just with a different, different textbook. Oh, hang on. Went too far here. Looking at one particular base, this is guanine. Lots of different problems that can happen to guanine due to living in these conditions. First, actually doesn't even show the depurination that happens here. Oxidation can happen on these bonds. Alkylation can happen here. Deamination can happen here. Alkylation can happen here. Lots and lots of different possibilities here. This, most common here, we're going to have deamination of cytosine. You lose the amine group here. What happens? Again, it's a hydrolysis reaction. You end up with uracil. Uh-oh, wait a minute. Uracil, normal base. Could be a problem, but as we'll see, because this is such a common occurrence, there are specific ways to get rid of these particular changes in nucleotides. Yeah?
So you said that's the most common one a few times now. So how am I going to get my mind in test mode for that?
So this one, actually the most common one is this one here. This one's slightly less common. So when I talk about the most common, it's going to be this one down here. Depurination. The most common mutation. What is the most common mutation that happens? So this is, exam question. Great. Okay. It may or may not be. If you look at old exams, I'm sure you'll see this multiple times. So second most common is this one. So the chemistry, however, is exactly the same. And I'm sorry about the figure down here at the bottom here. Hopefully you guys have it on your notes. The deamination that happens on 5-methylcytosine. And later we'll talk about 5-methylcytosine. Turns out it's a pretty common DNA modification that happens particularly in eukaryotic cells. If this gets deaminated, what happens is it turns into thymidine. And there's a problem with thymidine. If you have DNA damage, it generates thymidine. And that is the cell doesn't know that it's wrong. And in fact, probably a lot of the spontaneous mutations that happen in the human genome are exactly these kinds of mutations that happen and get maintained. It's because there is no easy way to get rid of this. I like this figure as well because it also shows this pyrimidine dimer which forms. Pyrimidine dimer, again, is covalent linkage between two pyrimidines that are right next to each other in the sequence. And because of this nice cyclobutane, it's a 4-bond structure between carbon atoms, That's a planar structure. So again, what you've done is you've really twisted the DNA to have these things lining up right on top of each other. Hopefully not beating the dead horse here. This is why I often have a question like this on exams, because we talk about it a lot. Is, again, this process, hydrolysis of, it's also known as the glycosidic bond here between your phosphodiester backbone coming off at the 1' position of your deoxyribose. Here's your purine that gets chopped off. Now you have a depurinated sugar, again just an OH here. Or second most common is the deamination of cytosine, which again is hydrolysis of this amine group leading to deoxyuracil, because it's still attached to the deoxyribose backbone here. So how do we deal with these kinds of changes? Smart organisms, i.e. unlike E. coli or humans, which have lost this gene actually, curiously enough, some organisms have a very specific protein that will correct pyrimidine dimers. It will bind to and recognize these pyrimidine dimers, which are formed due to UV irradiation, the cyclobutane ring, and through activation with light of a slightly different wavelength, will actually just completely reverse these bonds. It would be really convenient if we had one of these enzymes. Turns out we don't. Yeah?
So these are E. coli bacteria that have this specific...
Okay, so it turns out E. coli doesn't have photolyase either. The number of different bacteria, which if I told you I might ask you on an exam, which you probably don't need to remember. Yeah?
What does DNA photolyase do?
So DNA photolyase is it will bind to and recognize pyrimidine dimers, which have been formed by UV irradiation, and in the presence of longer wavelength light, lower activity, will just reverse that cyclobutane dimer. So it's a really efficient way of dealing with this kind of problem. Just now, unfortunately, we don't have that particular gene. So we have to have other ways of dealing with it. So we'll talk now about different kinds of repair. We talked about this one really briefly, but I think I did a really bad job because it was right at the end of the lecture and I was probably going far too fast. I mentioned mismatch repair. We already talked about polymerization, exonucleases as well, but the important thing here is really more about how these mismatches are taken care of. So again, it's the newly methylated DNA in, the unmethylated DNA that you have in E. coli, because it's the new strand which is being made. That's what's shown up here. And the mismatch which happens because the polymerase put in the wrong nucleotide, could have been a different tautomer. The proofreading enzyme didn't get to it. So you have a mismatch. This mismatch now has some kind of different structure. That structure is recognized by these mute genes and this binding here by the mut-S gene to, again, it's a structure. Here's a X-ray crystal structure of this mut-S enzyme bound to one of these mismatch bases. Because it's bound there, it will interact with a second protein, and this is a protein which is going to make a cut in the DNA. And I don't like this figure. It should be cutting on this side of this mismatch here. Because once it makes a cut there, now you've got a cut in the DNA on this side of your mismatch. You have a cut on this side. This cut could either be because the polymerases and ligases haven't got around to fixing it, particularly on your lagging strand, or these enzymes can make secondary nicks on either side of where this problem is. Then you have to have a helicase that's going to remove this new DNA that had this problem on it. And then we've got a template. We've got a 3' OH. This is perfectly good to go for any of the DNA polymerases inside the cell. So in theory, we talked about this before. In practice, I didn't think we did a decent job. So now let's talk about what happens with these other kinds of repair processes. We'll start over actually with B, so-called nucleotide excision repair. Nucleotide excision repair means big cuts, usually dealing with big problems in your DNA. And those big problems are things that can be detected by structure, and usually really easily detected by a change in the structure of the DNA. And I've been kind of harping on this whole thing with pyrimidine dimers. We're swishing the DNA. You squish the DNA, clearly a big problem, clearly a big change in the structure. That gets detected by various proteins that we'll talk about in just a second, and those then lead to cuts that happen in the strand that has that big change in it. DNA helicase will remove that big piece, and now you've got a gap, a template, a 3' OH. Now DNA polymerase and DNA ligase can fill that in. This is clearly also a pretty inefficient process because when you have one base change or one small-ish change, again with the mismatch repair, or here with these nucleotide excision repair, they're big changes. You have to repair above 10 nucleotides. If you think about each of those requires nucleotide triphosphate hydrolysis. It's a pretty inefficient process. On the other hand, the much more efficient process is the one over here in A, so-called base excision repair. And base excision repair also works for cutting out just one particular nucleotide in the backbone as well. It's not just the nucleotide base. So here's just the example of what happens with deaminated cytosine. Deaminated cytosine becomes uracil. Well, it turns out there's a very specific protein that basically scans along the DNA and says, hey, is there a uracil here? There's a uracil here. We're going to assume that there was a deamination event that took place. We're going to take it out and put in the right cytosine, no longer deaminated there. How does that happen? Again, there's a very specific protein called a DNA glycosylase. Remember I mentioned the glycosidic bond. The glycosidic bond is between the ribose and the base. So what does a DNA glycosylase do? It cuts that bond. So if you've got the wrong nucleotide there, that gets cut out by the DNA glycosylase. In this particular case, it's the uracil DNA glycosylase because it's cutting out uracil. Now you have a phosphodiester backbone missing a base. Now, this is a missing base. How else could you generate this? Depurination, depyrimidization would give you exactly the same thing. You're missing one base. This is not enzymatically, it's just regular hydrolysis that happens. So now, once you have what's called an abasic site, the AP endonuclease, which actually stands for apyridinic or apurinic, endonucleases, what do endonucleases do? They cut in the middle of DNA, not cutting in from the ends. The endonuclease will make a cut here. Then a phosphodiesterase, again, all these aces, what do? They're enzymes. They usually chop up what's right in front of them. So a phosphodiesterase is going to cut a phosphodiester backbone. And what this ends up with is you now have a gap of one base here, single nucleotide base, but you've got an OH, you've got a template, DNA polymerase will add your new nucleotide and reseal it. So this process here is a much, much more efficient process, but it depends on having something which is going to detect this one particular change that happens here. Yeah?
So the way I'm going to go home and study about this, I'm going to try to learn every step, right, and try to memorize everything, but that's not really the right way to go about this. So is there any kind of allosteric way of studying this that you could?
An allosteric way of studying this. That's a great terminology. Good question.
I don't want you to give away answers.
No. I haven't written the exam yet. I don't know what's going to be on the exam. So you can go back and look at the old ones. You'll see what I've done on some of these things. So we are going to go through many of these extra steps in the process that we go through here. So I would know the process here. And to basically say, okay, if we're talking about base excision repair, it's going to be a single nucleotide change. It's going to be a very specific one because you've got something that's going to recognize that. It's a very efficient process, but again, it depends on having one particular thing. The general process, which is in B, there, it's not dependent on having one particular change. It's something which messes with the structure of DNA. And so now it's a much more general process, but a much less efficient process. And we'll talk about, again, some of the enzymes to do this. One way that, and I've talked about a number of people about this already, one, I think, really good way of allosterically studying for the exam would be to take a blank piece of paper and write these things out, put the enzymes on them, label it. That's so what else people call concept maps. I think it's a great way of going through the process. Yeah?
I was going to ask, is excision nuclease just the go-to protein for dumb organisms that don't have that photolyase for getting rid of
Okay, so the question basically is, if you don't have photolyase, how do you do it? Yeah. And the answer is, you're exactly right, nucleotide excision repair. Okay, other questions on this? We will go through some more of the details here, but this is sort of the overview. And in terms of allosterically studying, this is a great place to start and then sort of fill things into that process. This is very much the overview. Again, we'll talk more about this as we move along here. So I mentioned before, basically, oh, sorry.
So going off of what you just said then, because let's say organisms that do have that
Photolyase?
yeah, photolyase, do they not then partake in nucleotide excision repair, or is that not determined?
Okay, so the question is, if an organism has photolyase, does it not have nucleotide excision repair? What do you think?
I think it probably does use it, but just
Why? Why would it be using it?
it's just another proofreading.
Because, well, these are the pyrimidine dimer formation is by no means the only kind of funky things that happen to your DNA. It's one of the more common ones, but certainly there are lots of other ones that happen too.
Okay, so this is the photolyse, photolyse, that's specifically for pyrimidine dimers, whereas these two mechanisms, the deaminated C and pyrimidine dimer, were just examples of mutations.
Yeah, exactly. And in fact, that's the one that we're moving to right now. Thank you for the lead-in. So deamination, again, this is the very specific process that happens. You have to have something that's going to detect that particular changed nucleotide, that particularly changed base. And the classic example here, of course, is uracil, the uracil DNA glycosylase. This particular one here is a structure of what happens to the DNA when you have one of these modified bases that's next to it, bound to the enzyme. And the enzyme is actually completely missing here. But that DNA glycosylase will recognize, usually, again, uracil, but there are a number of other kinds of deaminations that happen very frequently that you also want to have taken away. If you deaminate adenine, you have hypoxanthine. Do you think you might have a hypoxanthine DNA glycosylase? Because this is a pretty common mutation that happens. Yes, most organisms are going to have a hypoxanthine DNA glycosylase. Same thing is true for xanthine. No, I wouldn't expect you to remember the structures of all of these things, but you do have specific enzymes, which are the DNA glycosylases, that are going to recognize a lot of the very common kinds of changes which happen. Same thing is true with adeoxyguanine, that we'll take a look at in just a second here. Big problem again, as I mentioned before, 5-methylcytosine gives you thymine. If you had a thymine DNA glycosylase, what would happen? You'd chop out 25%, depending on what your genome is in terms of A's and T's, get rid of those bases. That would be incredibly inefficient. So you can't have a thymine DNA glycosylase, so you can't repair this kind of deamination. Let's take another quick look at this. Again, this is another one of those DNA glycosylases. This particular one is the adeoxo-G DNA glycosylase. Here's the protein up here in gray, the DNA down here in purple, with the adeoxo-G right here. So again, DNA glycosylase, what does it do? Chops the glycosidic bond. Now you have an AP site missing a base. AP endonuclease will cut on this side. Your phosphodiesterase will cut on this side. Now this will leave. You'll have a 3' OH opposite strand has the right template. Your DNA polymerase can fill in here, and the DNA ligase takes place from there. Need to change this. So that's base excision repair. Yeah?
Just on that last slide.
Yep.
Where it says AP endo-exo, is that because the enzyme itself can be endo or exo in its nuclease?
Okay, so endonuclease cuts in. Once you've made an endonuclease, now you have an end. And so an exonuclease can work on that end. Because you have to have the endo first, because there's no end. But once you have an endo, now you can have an exo, because there's an end that it can combine to and cleave off. So that order. Sounds like an exam question. What order would you have to have these in? Something to think about. Or maybe a clicker question, something like that. So that's base excision repair. Any questions on base excision repair before we move on to nucleotide excision repair? Nucleotide excision repair is when you have some kind of change in the structure. So again, up here, a distortion of the double helix. It could be a pyrimidine dimer, but it could be a whole bunch of other things, which is why you need another enzyme or the set of enzymes above and beyond photolyase. So this particular case, this is what happens in E. coli. In E. coli, you have the proteins called UVR A, B, and C. Why are they called UVR A, B, and C? Because if you make a mutation in these genes in E. coli, these are sensitive to UV radiation. So that's where they got their names from. Again, it's the geneticist. You break something, you figure out what it does, that's what you call it. So, sensitivity to UV radiation, what does that mean? What do you usually get with UV radiation? Pyrimidine dimers. So these are very important for nucleotide excision repair. That's how E. coli, because it doesn't have a photolyase, deals with these things. So, what happens? UVR-AB bind to the DNA. That then brings in UVR-C that will make cuts in the phosphodiester backbone on either side of whatever this change is. Then the UVR-D helicase will get rid of that DNA, and then our friends DNA polymerase and DNA ligase will fill that back in. One of the things that you'll notice here, once we have the binding to the DNA, where we have this change, there's also a big change in the rest of the structure of DNA. And that's, again, not critical exactly what is happening here, but changes in structure of DNA are really important for protein-DNA interactions. So if you don't have this change in structure, this bending, you're never going to have the interactions with the other proteins that happen here. We'll talk more and more about changes in structure of DNA. It's still normal base pairing interactions, but bent or twisted or something like that. We'll see this happening again and again. It's a pretty common theme that happens through the rest of the course. So this is what happens in E. coli. What happens in eukaryotes is very similar. They just all have different names. And I'm not putting up all these different names because I don't want you to worry about trying to remember them. Yes?
What happens to those two A's, red A's, at the bottom, at the bottom of the UVR?
UVR-A, so what's happening with UVR-A's? So UVR-A and UVR-B are this complex, and they do seem to be important for getting this whole thing to this mismatched DNA. But once you have this change in structure, UVR-A is lost. And basically, these guys seem to be a control over the activity of UVR-B. Because if UVR-B does this, that was interesting, does this process, see if we can get this to shut down, maybe not, ghost in the machine here, that if you don't have the dissociation of UVR-A, then UVR-B could be binding to too much DNA, and you end up with too many cleavages. So it's a regulatory subunit of this process. Again, don't worry about memorizing all of these things. Just that UVR-A, B, and C are what's important for the process. Again, when you draw up in your concept map, nucleotide excision repair, what happens? What are the things that you need to do it with? I wanted to mention one more thing, and this has to do with a phenomenon that people had noticed quite a long time ago, and that is that actively transcribed DNA, i.e. what you're using to make RNAs from, get repaired a lot faster than DNA which is normally not being transcribed. In the case of eukaryotes, this would be your heterochromatin. You have many more mutations that happen there that don't get fixed as fast as what's happening in euchromatin, for instance. This is the case in E. coli, but it turns out basically exactly the same thing happens in eukaryotic cells as well. And that is because transcription is so important, again, making these RNAs, that if the RNA polymerase runs into a problem, it wants to repair that problem, because it's clearly something which is being made into RNA. It's presumably now an important kind of protein which needs to be made. So what we now know happens is that the RNA polymerase is actually associated with these repair proteins. It basically brings them along for the ride in such a way that when there is a problem in a transcribed piece of DNA, it gets repaired faster than parts of the genome that don't have active transcription that's taking place on them. Again, this happens in E. coli, where you've got UVR A and B that are actually coming along for a ride with the RNA polymerase. Same thing is true for eukaryotic polymerase, particularly RNA polymerase II, that we'll talk about, way further on in the future. So that's it for our nice base excision repair, nucleotide excision repair, and preferential repair happening where you have actively transcribed DNA. Well, this is great. This is wonderful. Works well, and it takes advantage of the fact that you've got your redundant copy on the other side of your DNA. Well, what happens if you have a double-stranded break? Double-stranded break is a real problem because you don't have that extra copy on the inside. You've cut on both sides. So how do you know where to put that DNA back to? And this turns out to be something that happens again with ionizing radiation. Some of the chemotherapy drugs will lead to this. And again, it's really good because it leads to all of these checkpoints and so on and so forth. The other thing, and the other reason that people use radiation therapy and also to some extent chemotherapy, some of these chemotherapy drugs, is that some of the repair mechanisms, and particularly this first one, non-homologous end joining, actually end up leading to mutations. And we'll talk about how that happens in just a second here. Homologous recombination, this is taking advantage of the fact that, at least for us, we're diploid. So no, you don't have the redundancy on these strands, but mom comes to the rescue. So double-stranded break in the paternal chromatid, the maternal chromatid is right there. So it can provide a copy. And it turns out that this also happens in E. coli a lot as well. We'll talk more about homologous recombination on Wednesday. Interestingly enough, telomeres are double-stranded breaks, right? Because they're missing an end. So how do telomeres not end up leading to repair? And so maybe hooking telomeres together to give us finally circular chromosomes, which are so much easier to replicate anyway. So it turns out that a lot of the proteins that associate with these telomeres block what's happening with homologous recombination. And again, that's in the review article that I gave you a link to last time. Double-stranded breaks can, in many cases, actually lead to cancer. So with that break, you remember when we looked at the karyotypes? Remember there was one that had an extra piece of one chromosome that had attached to it, the Philadelphia chromosome. Leads to CML, which can be treated with Gleevec. That breaking and rejoining, that was double-stranded break repair. That was putting it on somewhere else. And so clear case of leading to cancer. But again, we've got our diploid case right here. Let's do the overview of double-stranded breaks, and we'll talk about non-homologous end joining, and then we'll finish for today because homologous recombination is totally cool and fascinating and incredibly confusing and hard to get through in a couple of minutes. So, non-homologous end joining. This is the quick and dirty way to deal with double-stranded breaks. What happens, again, you have a double-stranded break. Then the repair machinery, which we'll talk about in just a second, actually chops this DNA in from the ends a little bit, 10 nucleotides or so, and then pops those two ends back together. Now, clearly, this is a process which you're now losing genetic information. You've chopped those ends and then put them back together. So you're going to lose genetic information in that process. If you're an organism like us, where 98.5% of our genome is not coding an important protein, you can get away with this because you can put those ends back together. The junk DNA doesn't seem to make a huge difference if you chop it and then put those ends back together. On the other hand, homologous recombination gives you a perfect repair of this double-stranded break, except it uses the information from mom, for want of a better word here. We have a homologous chromosome right next to it. Again, we'll talk about that on Wednesday, but let's talk about non-homologous end joining right now, also known as NHEJ. Again, horrible nomenclature, but this is what a lot of people use. Double-stranded break in DNA. Very rare that your double-stranded break is actually going to be in phosphodiesters that are exactly across from each other. Usually they're offset a little bit, and exactly how that works with the ionizing radiation we can talk about, but it's not critically important now. Take radiation medicine if you're interested in learning more about that. But these ends are recognized by these proteins, the Ku proteins. Ku proteins, among other things, were discovered by a guy I brought my stereo from in graduate school. So have to talk about it. So what happens here is these ends are bound in a complex because if you let these ends float around, you can have translocation, all this stuff that happens again, these problems that you can have in cancer cells. These are all in a complex. The ends get chopped in a little bit, and then these get ligated back together. So again, it's a quick and dirty process. You have binding the ends, additional proteins, and the reason they're called additional proteins here means you don't have to remember them for an exam. And they then get put back together. This is just an X-ray crystal structure of the Ku enzymes here bound to this double-stranded piece of DNA. This non-homologous end joining, again, is a quick and dirty way of putting ends back together. But it also turns out that this little amount of mutation, a few nucleotides, plus or minus, actually almost always minus, turns out to be really important for generating antibody diversity. So one of the things that we really haven't talked about very much is changes that actually happen in DNA which are good for us. And it turns out that generating huge amounts of genetic diversity in antibody-producing cells and also other cells of the immune system, which make the T-cell receptors, again, the important thing is not all those details, but that having mutations that happen in your DNA in certain parts of your genome turns out to be extremely useful. And one of the reasons it's so useful is that antibodies have to have a huge amount of genetic diversity. And one way to generate that kind of diversity is through doing, it turns out, exactly the same process with exactly the same enzymes in those immune system cells to literally cause what's called hypermutation. So hypermutation happens in these immune system cells through exactly the same proteins and mechanism of this non-homologous end joining. And if you think about this from an evolutionary point of view, actually the adaptive immune system in antibodies is really pretty specific to vertebrates, and some vertebrates don't even have an adaptive immune system, but this adaptive immune system is a relatively recent invention, and probably because of this using of non-homologous end joining to put these two ends back together. So we'll stop here. We'll talk about homologous recombination on Wednesday.